Controlled drug delivery systems. Introduction. Controlled drug delivery system maintains suitable and desired drug release over an extended time period. A controlled dosage form is formulated using hydrophilic polymer matrix. An ideal controlled drug delivery system releases adequate amount of drug at regular time intervals and at the target site of action to maintain therapeutic levels of drug in blood plasma. The oral controlled drug delivery system offers potential advantages of controlled release rate and dose maintenance in plasma. The controlled release formulations are incorporated with swelling polymers or waxes or both for controlling the drug release rate. Reservoir system also provides the desired controlling release rate. Controlled release drug delivery system aims to reduce the dosing frequency, reduce the dose, and deliver the drug uniformly. Thus, controlled release dosage form releases the drugs continuously at a predetermined rate for a fixed time period, either systemically or locally to the target organ. Controlled release dosage forms provide better control of plasma drug levels, less dosage frequency, less side effects, increased efficacy, and constant delivery. Terminologies. Definitions. The related terminologies for controlled drug delivery or modified release delivery systems are as follows. 1. Controlled release formulation. The controlled release system maintains a constant supply of the drug at zero order rate by continuously releasing the drug for a certain time period in amount equivalent to that eliminated by the body. And controlled drug delivery system delivers ideal the drugs either locally or systematically at a predetermined rate for a specific time period. 2. Repeat action preparation. A drug dose equivalent to a single dose of the conventional drug formulation is initially released immediately after administration. After a certain time period, a second single dose is released. In some preparations, a third single dose is also released after a certain time. 3. Extended release formulation. This dosage form allows two-fold reduction in dosage frequency in comparison to the immediate release conventional dosage form. Examples of extended release dosage forms include controlled release, sustained release, and long-acting drug products. 4. Delayed release preparation. This dosage form releases a discrete portions of drug at a time or at specific time intervals after administration, although one portion may be released immediately after administration. An example of delayed release dosage form is enteric-coated dosage form. 5. Targeted release drug product. This dosage form releases drug at or near the intended physiological site of action. These dosage forms may have either immediate or extended release characteristics. 6. Site-specific targeting system. This system targets a drug directly to a certain biological location. The target is adjacent to or in the diseased organ or tissue. 7. Receptor targeting system. This system targets a drug directly to a certain biological location. The target is the particular receptor for a drug in an organ or tissue. Site-specific targeting and receptor targeting systems satisfy the spatial aspect of drug delivery and are considered as controlled drug delivery systems. 8. Controlled action system. This system provides a prolonged duration of drug release with predictability and reproducibility of drug release kinetics. The drug absorption rate is equal to the drug removal rate from the body. 9. Sustained action system. This system initially provides a sufficient amount of drug to the body to produce the desired pharmacological action. The remaining drug amount is released periodically and maintains the maximum initial pharmacological activity for a desirable time period in excess of time expected from usual single dose. 10. Prolonged action system. This system releases the drug over an extended time period during which pharmacological response is obtained, but it does not maintain the constant blood level. Rationale. The basic rationale for controlled drug delivery is to alter the pharmacokinetics and pharmacodynamics of pharmacologically active moieties by using novel drug delivery systems or by modifying the molecular structure and/or physiological parameters essential in a selected administration route. Thus, for designing optimum controlled release systems, a thorough understanding of the pharmacokinetics and pharmacodynamics of drug is required. However, if the doses are not administered at scheduled time, the resulting peaks and valleys reflect less than optimum drug therapy. For example, if a drug is administered at frequent doses, it will reach its minimum toxic concentration, MTC, and will also result in toxic side effects. If doses are missed, periods of sub-therapeutic drug blood levels or those below the minimum effective concentration, MIC, will result, with no patient benefit. The medical rationale for the development of controlled drug delivery system is important for the following aspects. 1. Reduced dosing frequency. Use of conventional drug therapy limits the frequency of dosing, i.e., a chance of skipping a dose. This problem can be resolved with the development of controlled release dosage form that releases the drug over an extended time period and maintains the desired drug concentration in blood. By increasing the time interval between the doses, the number of doses can be reduced from 3 to 4 times to 1 to 2 times. Proteins and peptides can be delivered into the body using a controlled release system. Tumor therapies can also be improved with enhanced targeting. Therapeutic efficacy and safety of drugs are improved by targeting the drugs in the body in a more precise manner, and by reducing the dosing frequency. 2. Reduced drug exposure to the biological environment. The CRDDS alters either the pharmacokinetic or pharmacodynamic profile of the active metabolites. Designing of such a system involves a property of rate controlling and achieving better plasma concentration. The CRDDS system controls the drug concentration in cells and tissues of the target sites. 3. Minimized plasma concentration fluctuations. With the conventional tablets or capsules, only a single and transient type of burst occurs, when the drug effect is above minimum effective concentration. The response is observed pharmacologically. But a problem arises when the response is narrow. In this case, CRDDS is used to reduce the fluctuations in plasma levels by retarding the absorption rate, accompanied by slower drug release. 4. Better patient compliance. Development of any dosage form aims to provide better patient compatibility. Designing a dosage regimen for a drug influences its duration of action, bioavailability, absorption rate, and elimination rate. These factors in turn influence the therapeutic response of the dosage form. The CRDDS are ideal for delivering the drug at the target site of the body over an extended time period, and thus for maintaining relationship between the plasma concentration levels and the therapeutic response, and for improving patient compliance. 5. Lower adverse side effects. The dose and dosing intervals of each drug designed as conventional dosage form are varied. Each drug produces a different therapeutic response in plasma concentrations. The unbalanced response results in improper therapeutic effect or undesirable side effects. In rate-controlled dosage forms, the dosing intervals are increased and fluctuations in plasma levels are reduced to minimize the risk of undesirable side effects. Incidences of side effects as a part of plasma concentration, and can be minimized by handling plasma drug concentration at a particular time. 6. Augmented efficacy. For designing an ideal drug delivery system, 2 prerequisites should be fulfilled. Firstly, the drug should be delivered at a rate that a body requires over a time period. Secondly, the action should be at the specific site at specific receptors. These goals can be achieved by using CRDDS. This system is biologically irresponsive to the external environment. The prolonged release dosage form slows down the absorption rate due to the slow release rate of the drug by small bursts over time, which improves the efficacy of CRDDS. By increasing the time interval between the doses, the total number of doses is reduced, thereby reducing the dosing frequency, and thus improving patient compliance.